Since ancient times, our planet have undergone many extinction events. Among all of these events, only a few creatures could survive, which are the microorganisms, specifically the bacteria. Different species of bacteria can adapt to different conditions, from very high temperatures like the hyperthermophiles, which can live to 90 degrees, to sacrophiles who can survive to -20. They can survive under different pH, lack of nutrients, and many other harsh conditions. All of that in an 8-micron microorganism. My name is Hazem Said, and today I'll be talking about alternative therapies for bacterial infection. And without further ado, let's get back to the track. Bacteria for humans is a double-edged weapon. Our body is an environment for many beneficial bacterial species, which help us in many things, like providing the first line of defense against other pathogens. They help us to digest food and contribute to the maturation of the immune system. Let me tell you that these bacteria are considered to be a part of our human genome, as if there is a deficiency in a type of bacteria that help us to digest food. That will lead us to digestion problems. Thus, this bacteria is vital for our health. On the other hand, certain species of bacteria are pathogenic, and in some cases, they can be fatal, like the famous pandemics through history, the plague and the cholera, which killed more than 20 million of people until 1928. When Alexander Fleming discovered the penicillin by mere chance. And that was the beginning of the antibiotic era, which is compounds made by many fungi strains that help us to kill bacteria by inhibiting its growth in many roots. But nothing lasts forever. The bacteria evolve many resistance mechanisms, like efflux pumps, which pumps the antibiotic out of the cell. They can decrease the permeability of their membrane to let the antibiotic enter. They can secrete enzymes that destroy the antibiotic or modify it, and many other ways to resist the antibiotic. So imagine if a single bacteria can possess all of these routes to resist antibiotics. We will have a super bacteria, aka superbugs, which can affect the human and cannot be treated with normal drugs. And let me tell you that we humans contributed to the rise of superbugs. How did we do that? By the excessive use of antibiotics without reason. This helps the bacteria to adapt to the drug and to share it between each other in a process called transgenesis, at which the bacteria can transform the resistant genes to another cell. Then we will get strains of bacteria which is super resistant to antibiotic. This problem became a global crisis by the WHO organization. But don't worry, my friends, because biotechnology always got the solution. If we look at nature, we will find an expert predator who cannot do anything in its life except for killing bacteria. I present to you the bacteriophage. The phage therapy relies on the use of normally occurring bacteriophage to infect and lyse the bacteria at the site of infection. Biotechnological advances have further expanded the potential of bacteriophages to include novel strategies like using bioengineered phages and purified phage-lactic proteins, which the phage uses to break the cellular membrane to enter the bacterial cell. The phage therapy is simple yet very effective. When the phage infects the cell, it starts to enter its DNA into it. The phage DNA makes the bacterial cell as its factory for making new phages. When it multiplies for a great number, it starts to burst out of the cell to find a new cell to infect. Actually, this is not really a novel approach. In 1915, bacteriophages were discovered by Frederick Thorpe, almost a decade before the discovery of penicillin. Phage therapy at that time were being developed to be a drug for bacterial infections, but at this time, they only knew little about bacteriophages. The discovery of penicillin after that was a revolution that stopped the researches which was made on the bacteriophage as a therapy. Though at this time, it gave many great results. 11 out of 12 mice was treated for bacterial infections using bacteriophage. Several children were also successfully treated with bacteriophage in hospital in France. Also in Belgium in 1921, patients with different bacterial infections were treated with the same way, and it showed great success after only 48 hours. Phage therapy have many advantages over the antibiotics. First, it's fatal for bacteria. Infected bacteria are unable to regain its viability. Low toxicity, as they are made from only proteins and nucleic acids. Host specificity. There are several types of bacteriophages. Each one of them can only target specific species of bacteria. Thus, it cannot infect our body cells or the beneficial bacteria in our guts. Unlike the antibiotics, which is broad spectrum, which means it can also kill the beneficial bacteria. Autodose. Phages can multiply their number in the bacterial cells, which makes the therapy transfer faster from bacterial cell to the other. Can be rapidly discovered. Bacteriophages are the most abundant bioentity in our planet. So can the bacterial natural enemy become our savior? Thank you for listening.